Aujourd'hui, on a le grand privilège et le bonheur d'accueillir Gregory Eaves, qui est professeur au département de biologie à l'Université McGill et qui est professeur en écologie aquatique. Elle est ici pour nous parler d'un grand projet, le projet Lake Pulse. Winnie a fait sa formation à Queens avec John Small, puis postdoc à l'Université d'Ottawa avec Jules Blais en toxicologie, travaillé partout au Canada, sur la côte ouest, dans le nord, au Québec. Et puis, elle s'intéresse à tout ce qui est l'écologie à long terme et puis les changements environnementaux planétaires. Winnie était aussi ma directrice de postdoctorat. Donc, je suis très contente qu'elle soit ici aujourd'hui et je vous souhaite un bon séminaire à tous.
Merci, Emily, et merci à Juan Carlos pour m'inviter et tout le monde de venir aujourd'hui. Je vais faire mon séminaire en anglais, mais je suis très à l'aise si vous voulez poser vos questions en français. Okay, so let's dive in. I had the pleasure of being a part of the Canadian NSERC Lake Pulse Network, which was a large sampling of lakes. And so I'm going to talk to you about the network, and then I'm also going to talk to you about projects that my students have done within the network. So the Lake Pulse Network started as this crazy idea that many scientists within the grill discussed and would not have happened without the fearless leadership of Yannick Huot, who was the director. But it was not just grill members. We brought in other scientists from across Canada. And really, this is just where the idea started, because what you'll see is that an impressive amount of fieldwork was done by students. And so I just want to shout out to a lot of the students. These are many of my students that were involved in the Lake Pulse Network. And then, of course, as we grew the project, there were many needs that we identified scientifically, and so we brought in a large number of collaborators. And so this is just some of the collaborators that I've worked with in the context of this project. So I am obviously going to talk about lakes. I'm sure with Warwick and Emily and Reinhardt in the crowd, this is not new to anyone, but I think lakes are really important ecosystems in general because of this very nice paper that Warwick contributed that argues that they're centers for integration, they're regulators, and they're important sentinels. There's many species that have this sensitive aquatic life cycle, and so changes in the species composition can tell you something about the changes to the environment. There's lots of biogeochemical reactions that are driven by water, so they're hotspots of biogeochemistry. And then finally, they're integrators, that lakes are low on the landscape and are collecting materials within the watershed and the airshed, as well as organisms that live in the lake. And many of these organisms leave some kind of signature in the sediment, be it a subfossil or DNA or a chemical signature. And so this is a nice paper put out by some colleagues at McGill who did a big exercise to look at where lakes are distributed across the planet. And you can see that in Canada, we have the highest concentration of inland waters. And so it was sort of unfortunate that up until 2016, there was no national standardized sampling of lakes in Canada. Canada has produced many prominent limnologists working in regional areas and doing great work, but no time in history had scientists come together and done this in a standardized way. So this is the motivation under which Lake Pulse was born. Another interesting motivation was the fact that we are dealing more and more with a very complicated web of drivers, human activities that are driving changes in our landscape in terms of all kinds of different stressors like salt intrusion, overfishing, temperature increases. You can pick your stressor of interest. And this has all kinds of impacts on aquatic ecosystems, from water quality to quantity, so on. And so we can make many different measurements and then link this to ecosystem services. So this is a complicated web. different drivers are acting in different regions. And so having a national scale lens allows us to identify which drivers are acting in which areas. That's not to say that limnologists in Canada haven't come together to do sort of meta-analysis work, and some of that has been very important. And one example is a study by Arihel and colleagues who brought together the microcystin data that had been collected by different ministries of environment and academics across the country, and definitely brought some new insight into microcystins. However, if you look critically at the data, you can recognize that the data were collected over a 10-year period. The sampling was done anytime between June and November, which are very different limnologically, and 4 different analytical and field methods were used. In addition, you measure microcystin as an agency often when you have a problem. So they detected in their almost 250 water bodies about a 41% detection of microcystins. Whereas in Lake Pulse, where we do a stratified random sampling, our detection is much lower. So it gives us a much more optimistic view of the state of lakes in Canada. So how was Lake Pulse organized? There were a number of axes, the largest of which, and what I'm going to focus on today, is known as the large-scale sampling of lakes. Okay, so over the course of between June and, end of June and early September, mostly July and August, if a lake was going to be stratified, this is when we went to 1 lake a day and measured many different parameters, which I'll talk about. We also collected cores from each of these lakes, although we did not keep all intervals of the core. I'll talk about that in a moment. So I am by training a paleolimnologist, so we look at these sediment archives, and in each lake we took what we call a top and a bottom sample. So the top 1 to 2 centimeters representing contemporary conditions, and the bottom sample a few centimeters below the puck. That bottom sample was picked at a strategic depth based on an analysis of the literature to know that it was pre-1880, pre-industrial conditions. Even if we had analyzed all top and bottom samples, that would have been something like 1,300 samples, would have buried any postdoc or student working on this. So the analyses that I'm going to show you today are based on a subset where we keep the original stratified sampling design. And we are working on full core work currently. Hamid Ganbari is a PhD student in the audience that I've had the pleasure of co-supervising. I'll show a slide from his work. But a lot of that work is ongoing because it's much more laborious. Okay, so in terms of how did we pick the lakes to be selected, is we decided to sample within what I call the Goldilocks zone. These are the very large lakes for which there are many existing long-term programs. For instance, Environment Canada sampling the Great Lakes. These very small lakes, if you go there in July, they might be dry. So we did not sample those lakes. So we're within this 0.1 to 100 kilometer squared size range. And then we did what we call a stratified random sampling design. For each ecozone, we looked at 12 different ecozones. We mapped out all lakes that were within 1 kilometer of the road, and then categorized them according to a human impact score, which is simply if you delineate the watershed and you classify the dominant land use in each pixel and score that according to the intensity of that land use, and then you take the sum or average of the score, that would give you a human impact score. So agriculture, for example, gets a very high score, whereas natural landscapes gets a very low score. So then in each of these blocks, we selected an equal number of lakes. So overall, we had 9 core ecozones where we did a more intensive sampling, whereas in our northern ecozones, we did a lighter sampling. And then we added a few additional lakes that partners had some special interest in. So if we look at the lakes based on the human impact and the ecozones that we covered, unfortunately, we did not cover much of the Arctic. We would have needed a budget about 5 times the size that we had, so that had to be let go. But you can see where there is intensive land use in Canada is between the Quebec City and Windsor corridor, where most of the population in Canada lives, and then also a pretty pronounced human impact signature in the prairies and boreal plains. So for each ecozone, we did that stratified random sampling design and used different thresholds for the lake selection, because had we used the same thresholds for what is a high impact in the prairies versus the boreal shield, we would have greatly undersampled the boreal shield. So this was just how lakes were selected, but subsequently in downstream analyses, I'm going to show you some analyses where people have categorized human impact, and there we've used the same threshold across the country. So if we just look at human impact as an index, we can see kind of what we saw before with the other map, but now we're seeing it on a continuous scale of human impact from 0 to 1. Again, showing much of the same pattern that we saw before, and these are our focal ecozones. Okay, so that sets up the structure of the project largely. I just realized I had a few more slides. One of the things that I think not only has this contributed great science, but I think it was an amazing training exercise for students. So teams of 4 to 5 students went out for 2 months with a truck and a boat and a bunch of money and went to 1 lake a day, every day, except on the day before shipping. They had 1 day off for shipping and relaxing, laundry, and then they were back out again. But these are the routes that they traveled all over the place, saw amazing parts of the country, talked to lake cottage owners, talked to all kinds of people who love their lakes in one way or another, and saw some of these landscapes. These are all pictures taken in BC, my home province, and you can see a huge heterogeneity in the kinds of lakes as well as their land use around the lakes. So what did we measure in a day at each lake? We tried to measure pretty much everything that you could measure in a day, but because you need several days to measure things like fish and macroinverts, those things we did not do. Although maybe we will do with eDNA. We're trying with some aspects of that. So anyways, I also wanted to highlight that we did, Yannick Huot is a remote sensing specialist, and so we did also take a lot of optical measurements so that we could calibrate measurements in situ with satellites, with the idea that that team could then scale up the study of 664 lakes to possibly all lakes in those ecozones. The network scientifically was organized into different themes. So I was most involved in theme 1, where, by, how much, and why are Canadian lakes changing in the Anthropocene. The second theme was more focused on indicators, planktonic, benthic, and microbial indicators, and which ones could be best used to measure different parameters of lake health. There was a remote sensing theme focusing on scaling up, and finally a last theme on ecosystem services and scenarios of environmental change going into the future. So this was an NSERC strategic network. I think it was among the last that were funded, and then we shifted to the Alliance program. But the call that we responded to was all about ecosystem health. And that actually gave us a bit of pause and debate amongst the PIs. Well, what is ecosystem health? How are we going to define it? And we dug into the literature. It has actually been a term that's been used since about the 1990s. And my group in particular has defined ecosystem health as comparing lakes within an ecozone along a gradient. So you can compare a more reference site to a more impacted site. That's more of a space for time substitution approach. You can also, with paleo, go back in time and look at lakes at the pre-industrial period versus today. And that is another way of looking at it, a reference point that controls for morphometry, for geology, and a bunch of other things. So I'm going to dive now into the student projects. And so we have looked at a variety of different parameters of aquatic ecosystem health. First, I'm going to talk about some basic water chemistry. Then I'm switching to sediment cores, so those are highlighted in brown. We've looked at algal and diatom assemblages in sediment cores. We've looked at cyanobacteria biomass and toxin concentration in the water column. And then moving up the food web, we've looked at zooplankton assemblages and diversity. We've looked at subfossil cladocerans. And then I'd like to just highlight some work that we've done recently, trying to look at a more holistic framework for biodiversity, looking at both the terrestrial and the aquatic component. So let's see how we do. Okay, so this is a first project that was done. Actually, Joe Sanchez-Chat was an undergrad student who approached me, and I said, you know, we're pretty late in the network at this point, and I was like, no one's actually analyzed the basic water chemistry data. We should do that. Joe, I think maybe this could be a good project for you. So this is a principal component analysis of the water quality data. The land use data has been plotted passively, and so you can see, though, that there is a close association between urban, ag, and pasture landscapes with higher ions and higher nutrients. And we also see differences in the ecozones that are color-coded here. And if we just extract the PC1 scores and plot them as a function of their ecozone identity, we can see indeed that the prairies and the boreal plains are distinct from the other ecozones. These are areas that are known to have a lot of agriculture and pasture land. And other studies have definitely shown that as you increase agriculture and pasture, that tends to lead to more runoff of nutrients. We also dove into the data by plotting percent agriculture as a function of some nutrients across different ecozones. And here, using this sort of ecosystem health lens of space for time, we can see that there are relationships here. But interestingly, we see that the prairie ecozone intercept is much higher than, for instance, the mixedwood plain. So this is a region that is naturally more nutrient rich. It's no surprise that farmers have done really well here. This is where most of our crops are grown in Canada. And if you dig back into sort of the paleo time, you can understand that there was actually a seaway that went in through much of central Canada. And so there are old marine sediments here. This is what is the underlying geology that allows for this naturally nutrient-rich landscape. So there's certainly a suggestion from these data that agriculture have enhanced this naturally nutrient-rich landscape, but it doesn't completely nail it, and this is where we need paleo. So this was work done by a postdoc, Catherine Griffiths, who looked at these top and bottom samples of the core and looked at the difference between the top and bottom assemblage and the diatoms by calculating a Bray-Curtis dissimilarity. So the more the assemblage changes from bottom to top, the higher score you have. And so she looked across all the different ecozones, and indeed you find that the prairies is the ecozone that has changed the most. So not only do we see across space that there is evidence that you increase nutrients as you increase agriculture, but also in this ecozone, we're seeing the greatest change over time. And if you just change the presentation of the data and look at the data as a function of low and high impact lakes, you indeed see that lakes that currently have the most intensive land use are the ones that have changed the most in terms of their diatom assemblage. So, we looked at diatoms. We also looked at chlorophyll A and its diagenetic products. And this was work done in John Small's lab, where he has a visible reflectance spectroscopy instrument. And so we can measure the chlorophyll A and its diagenetic products in the pre-industrial sample and plot that relative to the current one. And this data shows that 80% of the data are above this 1-to-1 line. So most lakes have actually shown an increase in chlorophyll A as well as its diagenetic products. We also found that most of the diatoms showed an increase in planktonic taxa across many, many lakes, suggesting that it can't just be an agricultural signal that we're seeing here, but rather something that is more pervasive. And this, we considered multiple hypotheses, but climate change seems to be the most logical one, where you have enhanced stratification and longer growing seasons, allowing for greater primary productivity. Well, so this came out almost 2 years ago, and Hamid Ganbari is PhD student who's been analyzing the long cores from the Lake Pulse lakes. We have about 83 lakes that are dated, and he's used hyperspectral imaging, a facility that's over in geography. And so this is every single data point. There's an impressive number of data points here of 90,000. So this is the GAM trend, distilling the trend across all of these cores. And again, we considered multiple hypotheses. We looked at precipitation. If you have more precipitation, that could lead to more runoff and potentially more nutrients, stimulating more primary productivity, but it really doesn't explain the trends in chlorophyll A that we see here. We also looked at grain size. You have changes in grain size in your cores if there's major erosional events. Again, that didn't explain the trends that we see. But temperature seems to explain it quite well. And so this is the GAM for temperature across all of these 83 cores. And he also did an analysis lake by lake, comparing the closest weather station data to the chlorophyll A. And most of these correlations were very strong. In fact, the ecozones are color-coded based on the average chlorophyll A to climate or temperature correlation. And all of these are bright colors, warm colors. So just to kind of sum up this first part of the talk, the water chemistry and diatom assemblage data shows evidence of land use impacts on lakes downstream. And we're seeing widespread increases in chlorophylls and diagenetic products, as well as increases in plankton over a really wide geographic area. And the best way to explain this is through changes in climate, particularly temperature, driving warmer growing seasons and longer growing seasons. Well, often when we have warmer growing seasons and more nutrients, we start to think about cyanobacteria, which is a key signature or symptom of aquatic ecosystem health. And I sort of started in this area of looking at large cyanobacteria data sets when the NLA, the US National Lake Assessment Program, released their data in 2007. And with a master's student at the time, we did an empirical analysis and identified that nitrogen and surface water temperature were the strongest predictors of cyanobacteria biomass. Admittedly, we don't explain all the variation. We're leaving almost 70% of the variation unexplained. And so we thought with Lake Pulse, this might be an opportunity where we have a much larger set of predictors that we could explain more of the variation in Canadian lakes. And so this work was done by Paul McKeegan, a former PhD student. This is what the map of cyanobacteria biomass looks like based on a snapshot sampling across Canada. And again, you can see that the prairies and boreal plains is lighting up in terms of high biomass, as well as a bunch of lakes in the Windsor to Quebec City corridor. And indeed, the prairies have higher cyanobacteria biomass than many of the other ecozones. We again did empirical modeling exercise using GAMs. And here we found that nutrients, again, in this case, TP, emerged as the strongest predictor. That has been shown in many studies, nitrogen or phosphorus. And in fact, if we used a linear mixed effect model, nitrogen also came out as a predictor, but we're penalized slightly by having a nonlinear model here. What's possibly most interesting from this analysis was that we found a positive relationship with Daphnia biomass. And a priori, we honestly didn't have a clear hypothesis as to how this would play out, because the literature is a bit of a mess. And so what we're sort of taught in limnology textbooks is that, as a classic eutrophic lake, when you have a lot of cyanobacteria, you tend to have very little biomass of zooplankton, because particularly zooplankton like daphnids that are filter feeders, their filtering apparatus gets clogged, and so you tend to see a reduction in Daphnia biomass. But there have been a number of papers that have come out over the last 10 to 20 years saying that, well, maybe there's an alternative view, and this is what we find support for, that, and we can't identify which mechanism is acting, but these are the possible mechanisms that could explain a positive relationship between cyanobacteria biomass and daphnid biomass, is that there are some studies that have found that daphnids can adapt to these toxins. We also found that where we found the greatest biomass of cyanobacteria tended to be in these prairie lakes that are shallow. They freeze over in winter. There's lots of biomass there that's still decomposing over winter, and so you can have winter fish kills because of total anoxia. And while there's cyanobacteria in the water column, there's also edible algae, and so some of the zooplankton can selectively graze on the edible phytoplankton that's still present. So that's where it is, and I think lots of interesting questions still remain to be evaluated here. We also considered the toxins of cyanobacteria, and the World Health Organization has sort of a way of classifying lakes according to the amount of cells per liter. So if we just looked at the cells per liter, this is what our map would look like in terms of risk when you recreate in a lake that has cyanobacteria. We're finding lakes in the prairies and boreal plains, as well as some in the mixedwood plains. But we did measure microcysteines using both ELISA, which is an immunosorbate analysis. And here we can see that, again, the highest concentrations were in the prairies and boreal plains. We were unfortunately not able to do that for this set of samples. But we also collaborated with a scientist at Environment Canada, Arthur Zastepa, who has an LC mass spec. So we were able to analyze the congeners of microcystin. Microcystins are actually made up of about 100 different chemicals that vary in toxicity, and then linked the toxin data to the phytoplankton data through a multiple factor analysis, which is what I'm showing here. And multiple factor analysis allows you to bring together different matrices. So we also looked at the environmental matrix and zooplankton. I'm just showing it on 2 panels for clarity, otherwise it's a giant mess. But you can see that many of the microcysteines are indeed associated with high nutrients and ions. There's some surprises too. Microcystin LR and LA are the most toxic congeners. They were the ones that were found most widespread. LA seems to be associated with more urban landscapes. Aphanizomenon and LR seem to be closely associated here, although aphanizomenon is not known to be a producer. So this is possibly something that needs further investigation. We're definitely identifying more and more taxa that are toxin producing as we look further and further. Another thing that Paul did during his PhD was an exercise to bring together 16S metabarcoding with the phytoplankton count data, with the idea of like, can we just go to 16S? Do we still need to be doing phytoplankton counts? And another student looked at 18S, which I'll talk about in a minute. So here he brought together data for about 400 lakes using the 16S, and then doing a ton of work on bringing these 2 data sets together. There was a lot of data curation that was needed. And so on the left panel, you're seeing the metabarcoding data. These bars are along a trophic gradient from oligotrophic to hypereutrophic lakes, and then the microscopy data here as cell counts. We also did it as cell biomass. It looks very similar. But so there's some striking trends that you see. One is that you definitely see some of these bloom-forming cyanobacteria, both in the microscopy and in the metabarcoding, just as you expect in the more nutrient-rich lakes. And the picocyanos, shown in this wine color, are more abundant in the oligo and mesotrophic lakes. These are all things that we knew before. But what's interesting is that there is an overexpression of picocyanos in the 16S. Truthfully, when I say microscopy, I'm talking about Lugol's cell counts, and Warwick is nodding and saying, you know, well, you shouldn't use Lugol's to do picocyanos, when he's absolutely right, that you often use a fluorescence microscope to count picocyanos. So it's not necessarily that the microscope is wrong, because maybe if we had also done the fluorescent microscopy, it would look a little bit more like the metabarcoding. What I think the biggest takeaway, though, is that there is decent congruence between the microscopy and metabarcoding. So if the goal of your study is to understand blooms, then probably the 16S approach is not too bad. And then for 18S, we did an analysis of about hundreds of lakes, I think about 400 lakes. And this was work done by a PhD student. Well, actually, she's now Dr. Rebecca Garner as of last week. And she did a nice modeling exercise looking at the among lake variation in the protist community and assigned trophic mode, either phototrophic, heterotrophic, or mixotrophic for many of the reeds. And we know for the phytoplankton, we would expect that physicochemistry would explain a lot of the variation, because we've been doing phytoplankton analyses for a long time. But the heterotrophs, the ciliates, like, we often don't study those, so we don't know a lot about them. And mixotrophs, same thing. And so her results for the phototrophs are more or less as one would expect. But I think what's interesting is what we learn about these groups that largely get understudied, the heterotrophs and the mixotrophs, quite interesting, are the least predictable, but these are the ones that have the most flexible life strategy, where they're sometimes photosynthesizing and sometimes taking in organic matter from the outside. So just to wrap up this chapter of the talk, for the cyanobacteria, nutrients are still the most important predictor, even if you consider a very wide range of predictors. We were able to improve on the amount of predictive power from 33 to 55, so that was a big step up. But I don't know that we're going to be able to do much better than that with snapshot samples. And actually, Marika Bolia, who did the NLA study, we also did a study of Canadian lakes where we had multiple samples over the growing season and then took the seasonal average. And there we were able to explain about 70 or 80% of the variation. So if you want to make really great models, then going multiple times over the growing season, because cyanobacteria are very noisy, would be the way to go. Good news in Canada is that actually most lakes don't have microcysteines, at least when you go once in the summer. So we can feel pretty good about the health of many of our lakes. And I think the congener composition data will further help refine mechanistic understanding of why and where cyanobacteria produce these toxins. And then finally, the metabarcoding data has been really interesting, particularly in those nutrient-rich lakes and being able to understand blooms. And the 18S data is going to expand our knowledge of phytoplankton and other microeukaryotes. Okay, I am going to now move on to the primary consumers, looking at the zooplankton. And I like this. This was sort of set the scene for us. Eric Jeppesen started as a zooplankton ecologist, and now he does many things. But he and colleagues wrote this nice paper because zooplankton got left out of the Water Framework Directive, which is sort of the lake pulse in Europe. They started much earlier. And he said this was a huge mistake because zooplankton are cost-efficient indicators and great indicators of ecological quality, and pulled together some existing data that he had to show this, looking at species richness trends of all zooplankton or just the crustaceans, shows a sharp decline in species richness as you go along a phosphorus gradient. Biomass also increases along a phosphorus gradient, and you see clear changes in composition. So this was a good sort of predictive lens for what we might expect to see in Canadian lakes, and indeed we find support. If you look at zooplankton or the crustacean biomass across Canada, we again see that where we see the greatest biomass is actually in those nutrient-rich prairie and boreal plain lakes. We then did a variance partitioning exercise to look at what are the different groups of predictors that can best explain composition. And we did have access to fish data. I should just explain this fish data, though, that this was done by another project known as Fish Hab that was funded to pull together the data from the Ministry of Fisheries across Canada and based on their presence. So we don't have biomass, and surprisingly, fish doesn't explain much of the variation in the zooplankton data. Whether it's a question of fish data quality remains to be determined, or whether when you're looking at such a large scale, whether water chemistry is just more important. That is something that we'll have to follow up in future work. So Cindy Paquette was the PhD student that looked at this, and she also did an analysis looking at functional traits of zooplankton, and we see very similar results. Cindy also looked at sediment cores, and so crustaceans, particularly the cladocerans, leave subfossils in the sediment, and so she looked at the diversity using a variety of different parameters from the pre-industrial to modern times. And so here I'm just showing you one example. And if this is no change from the pre-industrial to modern, and if it's above the line, then there are losses in diversity. And if it's below the line, there are gains in diversity. And she's binned all the lakes that she looked at. She looked at 100 lakes in Canada, whether their human impact score was low impact, mid impact, or high impact based on current land use. And what she found is that across all the different biodiversity metrics for taxonomy, that it was the high impact lakes that consistently saw losses, whereas the functional diversity was a lot noisier and less conclusive. So from the crustacean zooplankton composition, we found that this was most strongly related to water chemistry, although I think with fish data that was collected by ourselves or in a stronger state, we might have detected a stronger fish signal, but that remains to be determined. And the sediment cores showed that sites that have had the greatest land use change are the ones that have shown the biggest change in diversity. And then finally, Sufyan was a master's student who's just finished, and we had this idea of like, we've been very focused on aquatic diversity, but there's the whole watershed. And could we bring together remote sensing data that would allow us to estimate the diversity of the terrestrial realm? And how do these things compare if we want to look at preserving the health of the watershed? So Sufyan looked at the phytoplankton and zooplankton from Lake Pulse, and then used this source of data of tree species richness from the National Forest Inventory. And so what he did is with each data set, he took the Z score so that, for example, there are many more phytoplankton species than zooplankton. So not to off-weight the balance, everything was Z scored. And so this is what the total richness looks like across Canada when it's Z scored, where we see higher richness of phyto, zoo, and trees in Eastern Canada relative to Western Canada. And there could be multiple explanations. This is really a pattern that we've detected, but there are, of course, westerlies bringing migrants from west to east. And there's also a lot more precipitation in Eastern Canada. So these are some possible explanations. He also did an analysis known as a cascade regression tree, which he first forced to ecozone. So using ecozone, let's find ecozones of similar richness in terms of phyto, zoo, and trees, and then look at the influence of land use and what are the thresholds of land use that might explain changes that we see. So that's this analysis here. And so the distinct ecozone units that he's identified are the Atlantic Highlands and the Mixedwood Plains, and then all other ecozones were binned together. And so this is showing the Atlantic Highlands, and it's there's a threshold of percent agriculture in the watershed of 26%. And you can see that sites that have high agriculture tend to have a lower diversity in zooplankton and a lower diversity in trees. And then if we follow down this axis, this is the all other ecozones, we see a higher threshold of agriculture, and high agriculture resulted in lower species richness of all taxa. And when you can map that, what does that threshold of 44% agriculture look like on the landscape? And that's what it looks like in terms of its distribution. So this is a first attempt to try and bring terrestrial and aquatic folks together. There's still lots of work to do. We tried also looking at bird data, but the quality of the bird data was just not there to be able to do that. As you can imagine, we've collected many, many samples, and there's lots of projects still ongoing. David Zelke is a PhD student with Dermot Antoniades and myself, so we're doing a lot of work on sediment geochemistry and carbon burial, doing some work on core scanning using Pierre Francus's core scanner at INRS downtown here. We're also doing full core analyses of subfossil zooplankton and doing some 18S work looking at sediment DNA, and more to come. But I will stop talking now. I think across many metrics, most Canadian lakes are actually quite healthy, so we should feel really good about our lakes. I think we already did, but now we have some data to back that up. But where lakes have deviated most are in the prairies, the boreal plains, and the mixed wood plains. Equally important to the science is, I think, the way we have done the science. The trainees that have come through this program have cross-trained across many different areas and have become colleagues for life, I think. And finally, we have had the opportunity to apply many emerging methods. And so I think we're also pushing some analytical boundaries. And finally, a quick shout out that all the data as they're being published are made available. So all the basic water chemistry data you can download right now from the Gordon Foundation data stream, and our lab has a policy that we make all the data open access upon publication. So thank you so much for your attention, and I look forward to your questions. Peut-être en français.